Hello, everyone. Thank you for joining in on this webinar organized by PolyPlus. Today, we have the pleasure of having Dr. Reinhold Horlacher, who's the CSO of Trenzyme. We're going to start very soon, and we're just going to give a few more seconds to people joining in. Just to let you know how this webinar platform works. So you can, if you have questions, please feel free to already submit them by chat. And so there will be first a part where Reinhold will present, and then we'll have a Q&A session, and we'll address your questions. So we're going to wait just a few more seconds to allow everyone to connect in, and then we'll get started. Maybe what I can already do is give an intro, present Reinhold. So Reinhold is the CSO and Managing Director of Trenzyme, and he has a long track record as a protein production and cell line development specialist. So Trenzyme has 20 years of expertise in being a service provider for highly customized cell line development and recombinant protein production from development to GMP compliant production. And we've already in the past worked quite a bit with Trenzyme, and it's always a pleasure to have Reinhold share his expertise on protein production. So thank you very much, Reinhold.
Yeah, well, thank you very much, Alengo, for the kind introduction and for the invitation to talk a little bit about the topic of today, production of difficult to produce proteins, because this is our daily business. As you mentioned, we are a service provider for recombinant protein production, and usually the clients came to us with difficult target proteins, and therefore it's a pleasure today to give you a little bit insight into our experience and hopefully to give some hints how these difficulties can become overcome, and finally success with production of difficult protein is possible. I will take a look if it works. Yeah, perfect. So it works. So I will start with a quick overview of Trenzyme, and as Alengo mentioned, we are a service providing company since more than 20 years now, and we do cell line development and protein production. Protein production for R&D purposes, that means milligram protein and not kilogram. The company is a privately owned company, and our clients are mainly from the pharmaceutical industry, either biotechs, CDMO, diagnostic companies are also clients, and also research institutes and universities. Since many years now, we have quality management standards in place, and we are ISO 9001 certified. And to be able to offer more services in the highly regulated area like GMP, we rely on a special software solution, and this software, it's a LIMS type of software. This allows us the product identification and full process documentation and traceability during the processes. And as mentioned, this is mainly needed for cell line development in the GMP compliant area. So I will give you a very quick overview of the different services we are offering, and I don't want to go in detail. So on the right-hand side, you see these blue buttons. So this is our cell line business. It starts with the GMP compliant production cell line development, where we generate cell lines for production of pharmaceutical compounds like monoclonal antibodies or other proteins which are relevant for pharmaceutical application. The stable cell line development is more or less needed for research and development applications. So these cell lines are used by our clients for cell-based assays, like ion flux assays or other cell-based assays for compound screening. The third blue bottom, advanced cell services. This is even more sophisticated because we also do differentiation of induced pluripotent stem cells into different cell types. To our understanding, so these iPS-based compounds or cells are the new generation of screening systems because the cancer-based cell systems are not very close to the human body, and therefore either primary cells or stem cells derived systems are the new generation of screening systems. On the left-hand side, you can see the green bottom, and that's the topic of today, protein production and purification, and I will go into detail. So we offer also some catalog products, which are mainly catalog proteins, and I will give you a quick introduction at the end of my talk. And many of these catalog products are needed for diagnostic applications, and therefore we offer also, let's say, high quality catalog products, catalog proteins tailored for diagnostic applications. So, but now let's talk about difficult to express proteins and the strategies to increase the success rate. I think all of you participating today are familiar with producing proteins in different systems, and I think all of you are also familiar with the problems, with the issues. And one of them is that there is no production at all, which can happen and which also happens to us. And this is the case, for example, if the protein which should be produced is toxic, for example, for the host organism, or which also could be that there is a mistake in construct design, so on DNA level, that, for example, there is the promoter missing or whatever, let's say, artificial situation. This could happen, and if there's no production at all, I think it's always a good idea to look into the construct design on DNA level if everything looks okay. But it could also be that there is some degradation. So if you then look over the time, you may see some recombinant production immediately after transfection or after induction of trenzyme production in E. coli. And if you analyze the samples after a longer time, maybe all of the protein is gone and there is no signal, and therefore our second measure to do is after taking a look on DNA level to make a kinetic to look if the protein appears and then disappears afterwards. So these problems with related to no production at all, I think they are easy to figure out. Difficult or more difficult problems are if there is some production, but the yield is not sufficient for the desired application. So Usually you need a couple of milligrams to make your assays, and then you are happy. But sometimes you need gram amounts of proteins, and then the yield must be okay and sufficient, and you don't want to run many bioreactors to produce only 1 or 2 milligrams. So the yield is also very, very important. But if you see some expression, but the yield is not sufficient, this is a good starting point to increase the productivity, because there are many different possibilities to increase the yield. You have to, you should take care that the activity is okay. So at the end of the day, you need an active protein. For example, an enzyme should have the enzymatic activity, or an antibody. It should be able to bind that. Yeah. So I mentioned that if there's some production possible, but the yield is not sufficient. So this is a good starting point if the activity of a protein is okay. And then if you see good productivity, it often happens that you see no activity. So you, for example, can get aggregation. So this is a very familiar problem in E. coli, for example. So you have inclusion bodies, and the productivity is great, tons of proteins, but it's misfolded protein, and the protein is not active. Sometimes it's also important to have a certain post-translational modification, and if the expression host is not able to do this post-translational modification, for example, glycosylation is one of these post-translational modifications. If this is needed for activity, then you never get a proper activity by using, for example, a bacterial expression system. Sometimes accessory proteins are missing. So if proteins are only active by forming a complex, so then you will never get activity if you don't co-express the partners of these proteins. And then point number 4, sometimes proteins need cofactors like metal ions or vitamins, and if the expression host is not able to provide these cofactors, then you never get an active protein out of these expression systems. Sometimes it's possible to add these cofactors after having purified the protein, but sometimes it's not possible, or it's even not feasible to get proper folded protein without having the cofactors during the recombinant expression present in the system. So on the next slide, it's a lot of information on this slide. On the left-hand side, you see the amino acid sequence, so the letters of an amino acid of a protein. And if you're taking a closer look, you will see that this is the receptor binding domain of the coronavirus. And on the right-hand side, you can see the spike protein of coronavirus. It's a trimeric protein, and the receptor binding domain, it's the upper part given in red. So the way from left-hand to right-hand side, there is a lot of things to do for the expression system. And you will first introduce the DNA into your expression host. And from the DNA, there will be RNA synthesized, and from the RNA, the protein will be synthesized. by translation, and after having the protein available, there may be some assembly or processing needed to get a functional protein. And at each steps, there are some important considerations necessary. So on DNA level, you should think about genetic stability, because if you introduce the DNA in E. coli, or if you generate a stable mammalian production cell line, you want to have a stable system. So, for example, repeats or palindromes are not preferred, and you should reduce these repeats or think of methods to reduce them. Also, the GC content is of importance, and also homonucleotide stretches are okay if they are not too long, but if they are going to be longer, then they generate or they produce problems. The gene copy number is also an important consideration. So in E. coli, there's an easy method to regulate the gene copy number. You have low copy and high copy plasmids. So this is an easy method to control the copy number. In mammalian system, there it's a little bit more tricky to control the copy number. If you're going to for a stable production cell line, then you have integration of the genes into the genome, and there are some methods to increase multiple integrations, but the result could be that the cell line becomes less stable, because there are some recombination events resulting in a loss of DNA and loss of the information. So here the replication has to be considered, and the elements influencing the genetic stability during replication. If we go from DNA to RNA, so that's RNA synthesis and stability, there are also some elements you should consider. Also, again, here structural elements, because not only the DNA can cause some or could have some problems with repeats and hairpins, also RNA synthesis became more difficult or even impossible, and therefore, you should design your constructs well to have no negative structural elements within your RNA. There are also destabilizing RNA elements which could or which should omit it. You have, or there are some RNA translocation elements present. They are most likely in the untranslated region of RNA. These also have a big, or could have a big influence on productivity. Here again, repeats are not preferred, so if possible, omit repeats. And cryptic splice sites are also not good. And if you're going to do codon optimization by these tools which are available, for example, by companies doing codon optimization, they look for these splice sites and omit these cryptic splice sites. DNA methylation is also possible, since you know that methylation of DNA is a method to regulate down the expression level, and therefore you should also consider of DNA methylation. So if you look into the next step, so that's translation, the biosynthesis of the proteins. You have the initiation of the translation, and there you need some elements for the ribosome to start. So these Shine-Dalgarno sequence or Kozak sequence are important. And I think one of the most important parts is codon usage. So I mentioned already before, the codon preferences are different for different expression hosts, because there's this degenerated code available, and you should think of the expression host and then adapt the codon usage to the expression host of your choice. And this is nowadays, it could be done easily by gene synthesis, and afterwards you should have no problems with codon usage. If you're doing recombinant expression in E. coli, you can also do codon optimization, or you can use special strains. I think some of them are named codon plus, and they have the tRNAs for the rare codons. But these codon plus strains are the second best choice. I think the first best or the best choice is to do a proper codon optimization. You should also think about artificial ribosome entry sites. So it could occur that ribosomes not only starts at the beginning of the protein, but if there's an artificial entry site in between the open reading frame, that then the translation starts in the middle of the protein, which is not a good idea. And also RNA structure has some impact on translation, because again, if there are some hairpins or elements which are difficult to open for the ribosomes, then the translation stops at these positions, and you get a premature stop of the protein. And finally, the processing assembly step. This is important if you have a certain degree of cross-translational modification or targeting. So, for example, secreted proteins in mammalian systems, they need a proper secretion signal to be secreted into the media and to turn active. Many proteins also need folding helpers, chaperones, to have a proper conformation or proper activity. And also the position of purification tags could have an influence on the functionality of proteins. A simple 6-fold histidine tag, so only 6 amino acids positioned at the N-terminal part or the C-terminal part, could have a very deep impact on the activity of a protein. And if you have a rather complex proteins with different domains, so the domain composition have a deep impact on activity. And sometimes it makes sense to, for example, to only express a certain domain. And if your target protein is part of a complex with other proteins, it makes sense to co-express the partners to have a good activity of the target protein. So how to improve the success rate? I mentioned already many of different possibilities. But generally speaking, I think it's good to take a proven expression system and proven expression vectors, so that you're sure that the basics are functional. Then it's also a good idea to modulate the expression strength. So high expression is not always the best idea. Sometimes a lower expression level gives better results, and you can do this by taking different promoters. You can modulate the gene copy number, and it's also possible to work with RNA stability to increase or to decrease the expression level. And always use codon and DNA structure optimization. So I think this is the most important idea from today. And if you not succeed with producing a big protein with multiple domains, then try expression of specific domains only, if this is possible. Then modify the localization of an affinity text, as mentioned before. It's done easily, and the results are sometimes very, very good. And if you have a rather small protein or a domain, then sometimes it's a good idea to work with fusion proteins to fuse the target protein to a other protein which is easy to express, like maltose binding protein or GST. So there are several fusion partners out there, and you can choose different of them. And in some cases, it will give you good results. And it's also always a good idea to co-express these accessory proteins like chaperones or the complex formation partners. This makes it much more complicated, but finally it could give good results. So if you look into our tools we have here in a trenzyme available, so we play with many of them. So we have different expression systems. So we use E. coli, we use yeast, we use insect cells and mammalian cell lines, and we have a toolbox. We have more than 150 different expression vectors available, and these are continuously growing. And we pick them from our toolbox and clone the target into the backbone, and then we check for expression. In our toolbox, this is a simple cut and paste step, so we can subclone a target in all of our 150 expression vectors in the same way. We sometimes check up to 4 different host systems in parallel, but in most cases it's only 2 or 3. And you can see the list in the gray box. These are unique features. Sometimes fusion partners, secretion signals different, and we use these different features to increase the expression of difficult to express target proteins. And for sure, experience is always good, and we have more than 20 years experience, produced more than 1,000 different proteins for our clients, and this experience is it's always nice to have. So our process looks like this. So if on the left-hand side you can see the conventional host finding process, usually people start with the most easiest, with the cheapest one, which is E. coli, then may switch to yeast, insect cells, and maybe finally only succeed in mammalian cell lines. For difficult to express target proteins, we do this all in parallel. So we take our vector toolbox, we clone the target into different expression vectors for different host systems, and then do a small scale pilot experiments in these different host systems in parallel. And then, you will have the result in much shorter time and save a lot of time. So if you thinking of doing expression in parallel, then the number of parameters is increasing very fast. So here a simple experiment: one target in 4 expression constructs, and each construct in 3 strains, checking 2 different media and maybe 2 different growth condition or induction condition, will lead to a set of 48 parameters. So then it's clear you can't do this in shake flasks or other devices. You have to do it in small scale and in a parallel way. And we use microfermenter systems with plates. For E. coli, it's called biolector. For mammalian system, we have a special shaking device for these plates, and so this downsizing of expression conditions then will generate a lot of data at the same time, and you have to analyze these data, and then you are able to identify the best expression system and backbone and combination very easily. So here the process is shown. So we do gene synthesis, then we subclone the target into the different backbones, either E. coli, mammalian yeast, or insect cells. Then we do small-scale expression or transient transfection in case of the mammalian system. And for the transient transfection, we take for all our processes, we take factor PO from PolyPlus because it works greatly and produces very reproducible data in our hands. After having done the small-scale transient transfection or expression in E. coli, we will do also purification in small scale and then analyze the proteins by different methods. Usually it's an SDS page or Western blot. And after having done the high throughput small scale expression in 1 milliliter scale, we do an upscaling into 10 to 50 milliliters. And finally, we have our production scale, which starts at 0.3 liter and ends, in our case, with 10 liter vessels for E. coli or yeast system. And then we take the biomass and do the purification and the quality control. And again, at this level, we work with transient transfection in mammalian systems and can do transient transfection up to 20 liter scale. And we also rely on factor PO from PolyPlus at these high volumes, and it works great. And the upscaling from 1 milliliter to 10 liter is very easy with factor PO. So I will end with some case studies to give you some examples. So this is one difficult to express target protein. It's TGF-beta. And here on the left-hand side, you can see the structure of the TGF-beta protein. It contains a secretion signal. Our optimized or our modified TGF-beta has a his tag. It has a pro region, which is also called latency-associated peptide, given in yellow, and then the target TGF-beta in red. And the yellow and the red part, it's also called a small latent complex. And we analyzed the expression of the wild-type version with secretion signal A. And on the right-hand side, you can see the analytics by SDS page. And here you can see there's almost no expression of the target. By simply modifying the secretion signal, so the green part of the molecule, we get a pretty nice expression of the protein. So we were able to increase the productivity from 0.1 milligram per liter to 3 milligram per liter. And by modifying the construct by a special expression enhancer, which is, let's say, a fusion protein which will be cleaved off by these expression enhancers, so that's panel C, we were able to increase the yield to 15 milligram per liter. And yeah, the product, the TGF beta, is always the same in all three different constructs, and but the productivity was increased from 0.1 milligram to 15 milligrams per liter, only by optimizing the expression construct and the target. This case study, it's interleukin-2, also a very important protein, and here we have done transient transfection in HEK cells by use of Vector Pro, and here we worked with a fusion protein. So on the left-hand side, number one, that's the interleukin-2 without fusion partner, and you see a very poor expression. And by adding a fusion partner, which can be cleaved off afterwards, we were able to increase the expression level several fold, and the maltose binding protein is the fusion partner in this case, and it's cleavable, and this was a very big success to increase the yield of the target protein expression. Final case study, that's a viral protein. It's from African swine fever virus. Here we have tested different expression host systems, E. coli and mammalian. So on the left-hand side panel, or lanes 1 to 5, you can see the expression in E. coli. And so it's number one. It's difficult, a little bit difficult to see on the slide. So on the left-hand side, E. coli, and there you can see a lot of insoluble proteins. So these, a lot of inclusion bodies are formed, and almost no soluble protein was or could be purified. Then we used the fusion protein strategy in E. coli and generate a fusion protein. This is also on the left-hand side, given with the second green arrow. Sorry, I have no pointer here on to use. So it's the second green arrow, and this fusion protein strategy resulted in much more soluble protein, but still the results were not sufficient. And then we switched to a mammalian system. So we took the fusion protein strategy from E. coli and moved into HEK cells, and there we can produce. There we can produce a lot of soluble, properly folded, active P30 protein from African swine fever virus. And now we produce this protein for diagnostic purposes, and we deliver high milligram quantities to our customers, and we use the fusion protein strategy and expression in HEK cells and transient transfection by Vector Pro from PolyPlus guarantees the good results. Yeah, so at the end, if we have a new project, a new customer inquiry, we simply start with analyzing the protein sequence and then decide together with the client there's some consulting needed. If it's a standard protein, that means not difficult to express, then we offer guaranteed protein packages in either E. coli or mammalian. If it's a difficult to express or demanding protein, then we apply our modular system, our toolbox, and offer a customized protein production with custom tailored services. But at the end of the day, the client will receive the purified recombinant protein from Trenzyme. And mentioned in the beginning, we have also some catalog products available. For example, the P30 protein from African swine fever protein virus is such a catalog protein, or also the TGF-beta. And if you're interested, you can visit our web shop. The address is given here. And it's an increasing number of interesting proteins which are available on the web shop. It's easy shopping. You can pay by credit card, invoice, or direct bank transfer. You can choose different quantities, and if the quantity is not sufficient, you can ask us, and we can prepare also bulk quantities. So take a look at our web shop, and if there's any question, then feel free to ask us. These are some examples of catalog proteins. A newer one is the TEF protease. If you are working with proteins, so the TEF is a familiar protein to cleave off histex, for example. So we have an optimized TEF protease, which is fused to GFP. So it stabilizes the TEF protease, and it's also nice because it's green. And if you're interested, just take a look. We also have still the coronavirus proteins available, different variants, and this is still an important part of our web shop. Yeah, so here's some argument, some information why you should choose Trenzyme for your project. I mentioned before, our projects are highly modular. We have a lot of scientific experts working on the project. We have a very experienced project management team, always keeping you in contact or informed about the progress. And we are working for our customers since more than 22 years now. We are ISO 9001 certified, and if needed, GMP compliance is also possible, and ensures a very good quality. So that I will end my talk. Sorry for the problems with my camera, and I think it will not work for the rest of the webinar. So you have to take only my voice, not my picture. Sorry.
Thank you very much, Reinhold, for the presentation. And no worries, as long as we can hear you well and you can hear me asking the questions from the audience, I think we're all good. So we do have quite a few questions, so I'll start right away. There's a question regarding if you could give some examples of destabilizing RNA elements.
So these destabilizing RNA elements they're, let's say, regulatory elements the nature uses for high RNA turnover, and these are located in the 5' or 3' untranslated region. And if you are taking an existing expression backbone, so these expression backbones should not have these destabilizing elements. So if you want for a recombinant expression, it's a good idea not to take the endogenous RNA sequence to omit these destabilizing elements. I hope this will answer the questions a little bit. So there are different elements available. Hairpins and etc. So they are involved in regulation of RNA.
Thank you. So the following question is, at which scale do you produce in mammalian systems?
So we start with this test production in 1 milliliter scale, and then we scale up to 20 liters. So a single batch at Trenzyme at 18 liters. After having feed, etc., then it ends up with 20 liters.
Following question is, how efficient is the removal of the fusion protein? I think this was regarding one of the case studies you showed, where you had where you showed the immunoblot with and without the fusion protein. Yeah.
So that's a very good question. You have to check this in each case. Usually the tags could be removed, or the fusion partners can be removed easily, but there is no guarantee. So from time to time we have problems because the fusion partner and the target may buries the cleavage site, and then the protease is not able to have access to the cleavage site. So in principle, it works well, but it has to be checked. And we check this at a very small scale. We check it even in these pilots, 1 milliliter scale. If there's a fusion protein, we purify the protein, and then we do a one-shot cleavage experiment. And if it works well, then we are confident that it will work also in large scale.
Following next question is, do you have any recommendations or experience for expression strategies concerning membrane proteins, for example, transmembrane transporters?
Also very good question. So if you're going for membrane proteins, then it's, to our knowledge or experience, it's always good to lower the expression level as low as possible, because high expression level will cause aggregation problems, whatever. Then if you're going to express membrane proteins in E. coli, there are some tags or tags. I will name it tags available to guide membrane proteins efficiently into the membrane. So this is so we use this for E. coli, and it works sometimes well, sometimes there are still problems, but it's a good idea to check these membrane guiding tags.
And another question: if it's important to prioritize protein quality and homogeneity, would you ever use a simpler expression system over mammalian? If the aim, is protein quality and homogeneity.
So I think there is no golden rule. So I think for complex mammalian proteins, I would always go for a mammalian expression system. For rather small proteins, also for mammalian, if they are not secreted, then I will go for easier expression system like E. coli. So if it's a cytoplasmic mammalian protein, not too big, and then I will go first for E. coli, and then if it not works, then I will go to higher expression systems.
Let me see. There are additional questions. Which insect cell line do you prefer for greater protein yield? If you can share this information.
So the insect cells, we have so we do the construct design, and the production is done at a partner from us, and they have different cell lines they use. And they check different lines in parallel. So it's SF9, it's TNI and some other special cell lines from optimized from this company. So it's always good to check different insect cells in parallel. It's also difficult to name a specific cell line, because then the others are they work also, but this is also property of some suppliers. And if you're from academia, then it's easier. But if you're from industry, you have to consider IP situation. So this is also important to consider.
And we have time for 2 last questions. So, and any other questions that we haven't answered during the webinar, we'll reply to you by email. So last 2 questions. So first one is, do you have any particular recommendations for expression of proteins that are designated for crystal structure studies?
So if you're doing crystal structure, then I would prefer to do expression in E. coli if it works, because then you have no glycosylation, which could make some problems for crystallization. So if and bacterial expression host will work to produce the protein in a proper way, then I would prefer bacterial expression. But if expression will not work in bacterial, then I will go step by step into higher expression systems.
And the last question for today, Reinhold, is how many constructs or conditions do you typically analyze in parallel?
So I mentioned that we have these microbioreactor system. There we can analyze 48 in parallel. If we're going for mammalian, then we work with 24 well plates, and we can use multiple 24 well plates in parallel, ending up in usually 96 samples. That means we not do 96 different constructs, but sometimes 20 or 25 constructs in different cell lines, for example, CHO and HEC in parallel. So usually, it's around 15 to 20 different constructs per expression system.
All right. Thank you very much for attending this webinar. Thank you very much, Reinhold, for presenting and for taking the time to have a nice Q&A. So please feel free to, so this webinar will be on demand, of course, and you should receive an email, I think very soon, probably tomorrow, beginning of next week, for the on-demand link. And there you will have the slides, and you feel free to contact Trenzyme, feel free to contact us at PolyPlus. So we have, as Reinhold showed, dedicated transfection reagents for protein production. And we also have this plasmid engineering service. So please feel free to reach out to us. And I wish you a good end of the day.
Thank you. Goodbye.
Bye.